So today I will talk about a subject that we have been recently interested in at CILcare, which is the occurrence of sensorineural hearing loss in patients that have underlying chronic inflammatory disease. And we will focus particularly on three conditions, namely type 2 diabetes mellitus, chronic kidney disease, CKD, and inflammatory bowel disease, IBD, inclusive of ulcerative colitis and Crohn's disease, which altogether account for a global prevalence of approximately 1 billion people. And we will look at these conditions in the context of age-related hearing loss, where this natural process leads to loss of sensory cells and auditory neurons. But in these particular three conditions, it has been reported in the literature that they are at particularly increased risks of getting sensorineural hearing loss, at least 2 to 3 times higher risk. By taking the globally reported prevalence of hearing loss in these conditions, we can estimate a global patient size of several hundred millions of patients at risk. And they are also at risk of getting early presbycusis, as indicated in the case of IBD, where children and young adults have been reported with sensorineural hearing loss. So this presentation will be divided in three parts. The first part will cover the potential mechanisms that links these three chronic conditions, type 2 diabetes mellitus, chronic kidney disease, and inflammatory bowel disease, to hearing loss. In the second time, we will look at what possible therapeutic approach we can think in the future and in line with the current clinical trial landscape. And finally, we will look at different hearing assessment techniques that are best suited to monitor hearing in these patient subpopulations in order to get ready for clinical trials. So what are the potential mechanisms linking chronic diseases to hearing loss? Starting off with type 2 diabetes mellitus, we can report three main causes from the literature. The first one is called cochlear microangiopathy, and it is illustrated in this figure here, taken from a paper from 2021, comparing capillary vessels of diabetic patients versus normal capillary vessels located in the stria vascularis. And as you can see in blue here, there's a thickening of the basement membrane and a reduction of the capillary lumen. And this has a consequence of reducing cochlear blood flow and atrophy of marginal cells in the stria vascularis, which eventually has effect on the endocochlear potential. The second cause reported is low-grade systemic inflammation and oxidative stress that eventually leads to cochlear hair cell damage and loss. And it's illustrated here in this graph that has been published in 2006, where they looked at temporal bones collected from diabetic patients type 1, type 2 in gray bars versus control individuals in white. And what you can see is in the basal region of the cochlea, you have significantly higher losses of outer hair cells reported in diabetic patients versus control patients. And this is suspected to occur through mitochondrial dysfunction and inflammation related to hyperglycemia. And the final cause reported is peripheral auditory neuropathy. It can be illustrated in a way here published by Han et al. In 2018, where they looked at the number of synapses connecting to inner hair cells using CTBP2, which is a presynaptic marker that has been localized through immunofluorescence. And as you can see in diabetes mice versus control mice, there's a significant reduction in the number of dots, meaning a significant reduction in the number of synapses per inner hair cell. Also reported are a significant loss or decrease in spiral ganglion neuron cell density. And that accounts for the peripheral auditory neuropathy associated to diabetes. So as well in the literature, we find evidence that type 2 diabetes mellitus increases susceptibility to noise. This is mainly coming from preclinical or animal experiments where diabetic mice, when they are exposed to a noise causing normally temporary auditory threshold elevation, in this case, they exhibit a permanent increase in their auditory thresholds. As you can see in this figure here, reporting cross-section of spiral ganglion neurons, diabetic mice versus control mice, when they are exposed to the same level of noise, have a higher consequence in the reduction of the neuron's cell density. And finally, what we can also outline is the fact that the advanced glycation end product and receptor advanced glycation end product signaling in diabetic complications share common pathway to noise-induced hearing loss, namely the phosphatidylinositol 3K kinase AKT signaling pathway and the JAK-STAT signaling pathway, suggesting that there may be additive effects of inflammatory pathway caused by hyperglycemia and inflammatory pathway caused by noise, and that the consequences will be that there will be more damage at cochlear level. In terms of preclinical evidence, we report here some data published in 2021 by Liu et al., where they looked at a genetic model of mice, diabetic mice, that have been mutated on the leptin receptor gene, and that causes a type 2 diabetes phenotype, as indicated on the top left part of the slide, with associated obesity, increased body weight in diabetic mice in red versus control as heterozygous mice in blue, and hyperglycemia as well, with an increase in fasting blood glucose. And what they did in this paper is that they monitored the hearing function of the mice across 1 to 3-4 months of age, and they were able to show that there's an early onset and progressive hearing loss materialized by significantly increased ABR thresholds, especially at high frequency, 16 and 32 kilohertz, and that eventually at 14 weeks of age reaches 30 to 40 decibel shifts. They also point out synaptopathy evidence in this model as early as 6 weeks of age at the same frequency reported above, 16 and 32 kilohertz, and that is materialized by a reduction 2 to 3-fold of the wave 1 amplitude measured between diabetic mice in red versus heterozygous mice in blue. And that correlates quite well with observation as well on the number of synapses per inner cell, where they have shown that at 14 weeks of age, there's approximately a 30% reduction in the number of synapses. And finally, they also highlight the third cause that we mentioned before for hearing loss, which is microangiopathy, as confirmed by this comparison of cochlear blood flow, where there is approximately 50% reduction at 14 weeks of age in this type 2 diabetic mice model. In terms of clinical evidence, there have been a few studies, observational studies, looking at hearing function in type 2 diabetic patient. A recent one here in 2021 looked at a cohort of 157 patients, so 314 ears, and they've shown that 49% of type 2 diabetic patients aged between 30 and 60 years exhibit bilateral hearing loss. And what is interesting to note is that when we look at older patients that have longer duration of diabetes, the severity of the hearing loss increases. And we find more and more forms, moderate to severe, to moderately severe forms of hearing loss with the progression of the diabetic conditions. So as associated risk factors, it is reported that diabetes duration, typically over 10 years, or time-weighted glycated hemoglobin superior to 8%, are associated risk factor for hearing loss. It is also reported that type 2 diabetic patients have impaired auditory brainstem response profiles, and their profiles are mainly affected in the latency of the recorded waves. So here in this table, they have compared a group of type 2 diabetic patient versus a control group, and they've shown in all waves and also in interpeak between 1 and 3, 1 and 5, or 3 and 5 latencies, an increase, a significant, statistically significant increase in the latency and in the interpeak latency, highlighting what we have talked about before as the auditory peripheral neuropathy and also a central neuropathy. So now moving on to chronic kidney disease. What links this condition to hearing loss remains not fully understood and not very clear, with mainly two hypotheses formulated. One focus on inflammatory processes that may be caused by renal failure, and the other lies around a higher exposition to ototoxic compounds that would not be eliminated as well due to kidney failure. It's interesting to note that there are many similarities between the basement membrane of glomeruli and stria vascularis, and there may be an interplay as well, as mentioned before, between diabetes and chronic kidney disease, because we've seen that nephropathy is a common complication of diabetes that could have the similar consequences on the stria vascularis function that we talked about before. And there are many common genes and proteins that participate in the homeostasis of these two organs, the stria vascularis and the glomeruli. So there is an interplay that is not clearly identified, but clearly links chronic kidney failure with hearing loss. And it's typically illustrated as well in two syndromic disease, Alport syndrome and Fabry disease, that have been both rare disorders associated with both kidney failure and progressive hearing loss. Now, if we look at clinical evidence of chronic kidney disease and hearing loss, there are again a few studies that have looked at the occurrence, the prevalence of hearing loss, and it is very well increased in CKD patients versus age-matched control. But what is also interesting to note is that with increase or progression of the stage of chronic kidney disease from stage 3 to stage 5, this is materialized by a significant reduction in the glomerular filtration rate. The number of cases of mild to moderately severe hearing loss, again, strongly increases. So there is a link and an increased risk factor as well in CKD for sensorineural hearing loss with the disease stage, as well as also hypertension and hyperglycemia. Here below in the graph is reported the cumulative hazard as the disease develops in time, and you can see that in chronic kidney disease patient, the risk in the end after 9 to 10 years of disease is actually more than doubled to those of non-CKD patient to develop sudden sensorineural hearing loss. And there has been a study as well in this paper of Meniere's disease, which is also a little less but significantly increased in the case of CKD. Now, in terms of preclinical evidence, there is no clear available model to study hearing loss in association to CKD in animal species, but there could be some that are put in place, especially one using streptozotocin treatment in rodents. This has been shown to induce diabetic nephropathy, and what could be done is to monitor the hearing function of these animals in time and investigate how progressive and how early the hearing function is affected. Similarly, we could also use a genetic mouse model of Alport syndrome. It has been shown to exhibit stria vascularis basement membrane abnormality, as reported before, and that it involves as well gene expression patterns for inflammatory processes, for example, the tumor necrosis factor alpha, that are also common to noise-induced hearing loss, which highlights again a possible interplay and additive effect with noise exposure in those CKD patients. So there could be a way that we monitor hearing in this genetic mouse model of Alport syndrome, and that we maybe combine it with noise exposure at low but regular levels to determine if it has a higher impact on the hearing function. So now coming to inflammatory bowel disease and hearing loss, What are the links? Again, they are not very well understood, but two are mainly put forward. Again, one caused by systemic inflammation, and here is illustrated by this figure taken from a paper from 2021 with a proposed gut inner ear axis that would result in sensorineural hearing loss. It all starts with dysbiosis at the intestine level, causing systemic inflammation, and then through a blood labyrinth barrier bridge, this inflammation will diffuse into the cochlea and may have consequences on inner and outer hair cell function. The second hypothesis lies around an autoimmune inner ear disease associated to inflammatory bowel disease, and that would cause a rapidly progressive bilateral sudden sensorineural hearing loss as a consequence of the inflammatory bowel disease. So in terms of clinical evidence, again, there are a few papers, quite recent, that have reported a much higher prevalence of sensorineural hearing loss in IBD population. We are talking, depending on the studies that we are looking at, between 38% to 60% of the IBD population being concerned by sensorineural hearing loss versus 14% approximately in control age-matched group. So there is a statistically significant increase of risk to get complication in the inner ear from inflammatory bowel disease. In terms of preclinical evidence, again, there is no clear available model to study hearing loss in association to this chronic condition. But again, we identified possible model of dextran sodium sulfate induced colitis in mice, because this model was shown to induce a rapid chronic ulcerative colitis symptoms, especially in C57 black 6J mice, and through activation of inflammatory processes, again, the phosphatidylinositol 3K, AKT, or NF-κB signaling cascade, leading to colonic inflammatory processes. And these processes are also found in noise-induced hearing loss mechanism. So there may be, again, an interplay and an increased susceptibility to noise in this mouse model that would be interesting to monitor and to evidence. So now that we have highlighted the different mechanisms that may link this chronic disease to hearing loss, what could be the therapeutic approaches that we can think of in the future? Well, in this table, we have summarized mainly three treatment approaches. One is neural protection. Another one is hair cell protection. And finally, the last one would be the hair cell regeneration. And taking this first example of neural protection with synapse repair and spiral ganglion neuron preservation, this would typically tackle the peripheral auditory neuropathy that we have reported in the case of diabetes mellitus. And for this particular approach, we can obviously think of the use of neurotrophic factors. Now, when we come to hair cell protection, there are two different angles that we can look at. One would be the use of anti-inflammatory and antioxidant drugs that would be maybe particularly suited to tackle the inflammation associated to diabetes mellitus and chronic kidney disease and protect hair cell from these inflammatory and oxidative processes and preserve them. When we look at the angle of inflammatory bowel disease and associated autoimmune inner ear disease, we can maybe more think of anti-inflammatory and immunosuppressant combined activities that we can find in, for example, glucocorticoid treatment. And finally, hair cell regeneration is a therapeutic approach that will aim to differentiate supporting cells into new hair cells, and that would be particularly suited for late stages of diabetes mellitus and chronic kidney disease, where we think that typically inner and outer hair cells might have been already damaged, and the patient could benefit from regeneration of these sensory cells. We can summarize the therapeutic approach that we've mentioned before using this graph published in 2021 with a global pipeline of compounds or candidates developed in treatment of hearing loss. And what we can see is that all the candidates that are reported for sensorineural or noise-induced hearing loss or age-related hearing loss, autoimmune ear disease, and Meniere's disease may potentially be also used for treatment strategy to address hearing loss in the chronic inflammatory disease that we've mentioned above. So there is quite a fair amount of compounds that maybe would be interested to test in the sensorineural hearing loss occurring as a complication of these chronic diseases. Despite the fact that there are many drugs or drug candidates that would be interesting to test, the current clinical trial landscape is actually quite poor. And when we look at clinicaltrials.gov and search for hearing loss in association to diabetes mellitus, for example, only 21 studies are coming up. They mainly focus on maternally inherited diabetes and deafness or Wolfram syndrome. And there's perhaps one interesting trial to mention, which is looking at the preservation of auditory function in Wolfram syndrome using valproate treatment. When we look at chronic kidney disease or inflammatory bowel disease, it's even worse with only 5 studies. And all studies are actually large observational studies looking at rare diseases and not studying their relation at all to hearing function. So we believe that this subpopulation of patients could actually be used as a defined set of patients and are particularly suited for carrying out observational in the first place and then interventional trials, maybe with drug candidates that we've mentioned before, that would have been selected based on their mechanism of action compatible to the pathophysiology. So now moving on to the last part of the presentation, we'll talk about hearing evaluation techniques that will be particularly suited to monitor the hearing function in patients suffering from chronic diseases. So obviously we can use the 3 most standard techniques, the pure tone audiometry, the auditory brainstem response, or the distortion product autoacoustic emission to monitor hearing and to use them to investigate efficacy of drug treatment in clinical trials, but there may be some limitation. Actually, with regards to the pure tone audiometry, it's reported that the early presbycusis starts with an increase in audiometric threshold at high frequencies. And with the standard range of 250 Hz to 8 kHz, maybe it will not be sufficient and sufficiently adapted to detect early signs of hearing loss in patients with chronic inflammatory disease, as it may start with a frequency above 8 kHz. With regards to click-evoked ABRs and distortion product autoacoustic emission, they are both useful, either to investigate rapidly audiometric thresholds and peripheral neural impairments through ABRs or function of outer hair cell through DPOAE. But the problem is more lying around equipment standardization that could be improved to lower variability, especially when looking at evaluation of patients across different sites in a single study. So we will now talk about extended high frequency and why we think this test is actually very well suited to monitor hearing in a patient with chronic inflammatory diseases. It typically looks at hearing thresholds at frequency above 8 kHz and up to 16 kHz, and is therefore very useful for detection of early presbycusis, as we've said. And there's a nice example of that coming from a paper from 2020, where they looked at pure tone audiometry through 250 Hz up to 16 kHz between a patient group consisting of children with inflammatory bowel disease versus a control group. And what you can see is that only at frequencies above 8 kHz, there is a statistically significant increase in their auditory threshold. So by using standard techniques, focusing on the 250 Hz to 8 kHz, this diagnosis might have been missed. And I will finish with this technique by saying that it's now more and more available on mobile equipment using headphones and tablet-based application. And that makes it quite readily available for clinical monitoring. So there really is an interest in pushing to use such technologies to monitor early presbycusis in chronic inflammatory disease. Now, another interesting test that we can mention is speech-in-noise tests. These are useful tests because self-reports of hearing and understanding difficulties in background noise are also very early signs of presbycusis and synaptopathy. So this speech-in-noise test, what they do, as illustrated in this figure, is that they evaluate the number of correctly identified speech. Can be words, it can be digits, it can be sentences, reported as intelligibility on a percentage scale, varying across different signal-to-noise ratio that are proposed, that are used to propose the speech. And comparing two curves, in purple, the normal hearing curve, and in red, the speech in noise impaired curve, you can see that there is an increase in what we call the SRT 50, which is the speech recognition threshold 50, the level of signal to noise ratio at which the patient is able to correctly identify 50% of the presented speech. So naturally, through impairment, there will be an increase of this speech recognition threshold. But there also can be a decrease in the maximum number of correctly identified speech. That's also a different sign. And also the slope of the curve may be indicative of an impairment in terms of speech intelligibility. Unfortunately, not all the speech in noise tests are equivalent. We can cite two tests that we think would be mostly suited for monitoring hearing in chronic inflammatory disease. One would be a validated digit in noise triplet test presented with headphones and tablet-based applications because it seems to be a fast and reliable method for screening. As you can see here, they looked in this paper from 2021 at the correlation between the speech recognition threshold and the audiometry, and they found that patients cluster very well with normative values for normal hearing at around minus 15 decibel in signal to noise, and that progressively, when we look at mild to moderate hearing loss, there's an increase in the speech recognition threshold from 12 to 10 decibel. And this test can easily be implemented in clinical trials to screen and include patients that have a speech in noise impairment. Now, when it comes to hearing efficacy assessment and evaluation of treatment, it seems that the matrix test appears to be the gold standard or the excellent test that should be used for three main reasons. It is universal, it is validated in several languages, and it's also designed to limit memory effect, which can be very useful when repeating the test during an evaluation period of patients where you repeat the different tests and you limit the habituation of the patient. Illustrated here are the performance of several tests with the recommendation of French Society for Audiology. And in green here is the performance of the matrix test with a minus 6 decibel normative value for signal to noise ratio. And we can see a fairly low dispersion of variability across this normative value. So as a take-home message, I would say that patients with chronic inflammatory diseases have significantly higher risk of sensorineural hearing loss, and that their hearing should be monitored as part of the daily management of their chronic pathology. This is more and more being raised by organizations such as the Center for Disease Control, who has recently published recommendation for monitoring hearing in diabetic patients. We think that type 2 diabetes, chronic kidney disease, and inflammatory bowel disease patients constitute a subpopulation of choice that could boost clinical research in hearing. But for this, there's the need of more observational studies that could aim to characterize their hearing profile and help standardize the set of hearing evaluation techniques that are used to advance pharmacological candidates into clinical trials. Thank you for your attention.
Nickel.